Thank you for asking me to talk. Um, I'll be talking today on genetics in dystonia in 2023 as part of September Dystonia Awareness Month on behalf of Dystonia Network Australia. My name is Kishore Kumar. I'm a neurologist. This is my disclosure slide, and I'd like to disclose that I received funding from the Paul Ainsworth Family Foundation to study dystonia, as well as the Medical Research Future Fund grants and grants from the Global Parkinson's Program. This is a bit of an introduction. I work at Concord Repatriation General Hospital in Sydney, in genetic testing lab and also the neurology department. And I work at the Garvan Institute of Medical Research as a group lead over there. So I'll give you a bit of an overview of dystonia in the genetics of dystonia, and then more of an update on some recent important developments. So here we have this kind of categorization of dystonia genetics according to the person's clinical picture. For example, if they just have dystonia, they may have one of these genes. If they have dystonia plus some additional movement disorders, such as features of Parkinson's or jerky movements called Myoclonus, they may have these other genes. So this is kind of a way of thinking about dystonia genetics. It can be different according to the clinical picture. One of the first causes of dystonia that we've identified was the TOR1A gene. And this is a common cause of dystonia, tends to start lower in the body, progresses upwards, involves the limb and the torso with general sparing of the face and neck. It also has low penetrance. This means that you can get the mutation but not get the disease. And it can change according to the age of onset and which sites are involved. And all patients generally have this mutation, which is a deletion of these three letters, GAG. More recently, we found dystonia caused by a mutation in this gene, GNAL, and this tends to cause dystonia that affects the neck and the head region, begins in the fourth decade of life. I did a study while I was in Germany and identified several patients, including this patient. And you can see here that he has tilting of his head to the left. This is a feature of his craniocervical dystonia, and identified a mutation in the GNAL gene in his case, and did some laboratory studies to confirm that this gene was indeed causing his dystonia. This is a really new and important cause of dystonia called the KMT2B gene, which causes early onset dystonia that can become generalized and is probably the most common cause of dystonia outside of the Ashkenazi Jewish population. Here's a patient with KMT2B dystonia that we diagnosed with genetic testing. He actually has a silent mutation, which means the letters of the protein don't change, but the way that the gene is put together or spliced is changed. And we did laboratory studies to prove this. Fairly recently, we discovered this new and important gene causing dystonia called the VPS16 gene. And this is involving a new pathway for dystonia involving a complex known as the HOPS complex, which and if you can see here on the fibroblast biopsies, the patients have this change on, if you look on a fibroblast biopsy, with this change in the lipids. And this is a feature of VPS16 causing dystonia. So here we have one of our patients who has this type of dystonia, VPS16, as part of this original gene discovery study. And he has these abnormal movements around the face. Here is another patient who has a different mutation. These patients are not related, but you can see here that she has these abnormal movements around the mouth. and her speech is affected. So this is a new type of dystonia called DYT, dystonia, DYT, VPS16 gene. Then we have another category of dystonia called combined dystonia, when the dystonia is combined with another movement disorder. And this is, and in some cases, dystonia can be combined with Parkinsonism. And here are some of the common causes, and I'll go through a few of those. One of them is called the GCH1 gene, which is the commonest cause of dopamine-responsive dystonia. When it's a single, when it involves one copy of the gene, it's called Sagawa's disease. So these patients have a good response to treatment with a drug called levodopa, which is the same treatment we use for Parkinson's. Here's one of our patients who has, as you can see, his head is tilted to the right. So he has cervical dystonia. This is him without any treatment with levodopa medication. And his movements are slow. This is called bradykinesia. So the dystonia is teamed with a Parkinsonism. And once he gets treated, his dystonia is much better. So he's taken this treatment called levodopa, and his dystonia has improved. This is dopa-responsive dystonia. And here we diagnose the case with the very advanced genomics called whole genome sequencing and long-read sequencing. We found that part of the gene had become detached and flipped around, and that copy of the gene was not functional. So this is called an inversion in the gene. I'd like to mention another form of dystonia, which is quite rare and unusual. It's called X-linked dystonia Parkinson. So this cause of dystonia is on the X chromosome, and it comes from this island at the Philippines called Panay Island. And again, this has a combination of dystonia and Parkinsonism, and it mainly affects males. And this is a patient who has X-linked dystonia Parkinsonism, and he's about to go for a special treatment called deep brain stimulation, and that's why he's wearing a surgical cap. So you can see he's got dystonia, severe dystonia of the legs. And he's also got dystonia of his trunk. And finally, I think it's important to mention this one. It's dystonia caused by a mutation in a gene called Parkin. This is the commonest cause of early onset Parkinson's disease below the age of 40 years. And many patients present with dystonia in the legs. So you can see here is one of my patients with dystonia caused by a Parkin mutation, a gene that really is most well known to cause Parkinson's disease. And she has this terrible cramping, twisting of the foot. So some Parkinson's disease genes also cause dystonia. And we can also get dystonia combined with irregular jerky movements called myoclonus. Here we have dystonia due to a mutation in a gene called the epsilon sarcoglycan gene, or SGCE gene. So here we have the patient having pulling back of the head, cervical dystonia, and you'll notice that he has these irregular jerky movements. And here we use a special technique looking at the whole genome sequencing data to show that he has a missing part of this SGCE gene. Another form of dystonia that I think is really interesting to mention is dystonia caused by a mutation in this gene, the TUB4A gene. In this case, the dystonia typically involves the person's voice and gives them a whispering voice. Here we have a patient who has dystonia, which is severe, involving their face and affecting his swallowing. And here you'll see a classic feature of the dystonia. touch your ear and touch my. Stick your tongue out. He's having trouble with his tongue movements. Lovely yellow lyrics. You hear his voice, he's got a whispering voice. And this is the type of dystonia that was identified in patients in a family from Queensland. This is another really interesting and new form of dystonia. This person has this sudden burst of dystonia that occur at night time, called paroxysmal worsening of dystonia. But it has generalized dystonia, and he has a mutation in this gene called the ADCY5 gene. And the interesting thing about this gene is that the pathway, by looking at the pathway, we know it involves caffeine, and you can actually treat patients by getting them to drink caffeine and they can improve. So this is a new development, the idea that a certain type of dystonia responds to caffeine. Finally, one of the really important things that we've observed is that different genetic forms of dystonia have respond differently to deep brain stimulation. So some forms, like the TOR1A, the X-linked dystonia Parkinsonism due to this gene TAF1, the SGCE gene, GNAL gene, and KMT2B gene respond really well to deep brain stimulation. Others we don't have a lot of evidence for, like the VPS16 gene, and there appears to be some that don't respond very well at all. For example, the THAP1 gene. So dystonia may be important. Genetic testing in dystonia may be important, and it may be particularly important if you're considering going for deep brain stimulation. And just to give you an idea about what you might are likely to expect from the procedure. But there are all sorts of other reasons why genetic testing in dystonia can be helpful, such as genetic counseling, family planning, and getting an idea of what might happen in the future. This is your prognosis. So thank you for this short talk today. And I'd like to thank my collaborators all across Sydney and also in India as well, and particularly Concord Hospital, Michael Hayes, Stephen Tish in St. Vincent's Hospital, Professor Victor Fung, and Dr. Neil Mahant at Westmead, and Professor Carolyn Sue at Neuroscience Australia, Research Australia, and my mentor in Germany, Professor Christine Klein. And again, thank you so much to the Paul Ainsworth Family Foundation. And thank you for Dystonia Network of Australia for asking me to talk. Thank you.